Scientists have just achieved a groundbreaking feat, creating human eggs from ordinary skin cells. This discovery could transform fertility, redefine reproduction, and open doors to possibilities once thought impossible. Today, we dive into science, ethics, and the future of life itself. The breakthrough explained. Scientists have recently achieved a remarkable breakthrough by creating human eggs from skin cells. This discovery represents a major step forward in reproductive science and opens new possibilities for people facing fertility challenges. Using advanced cell reprogramming techniques, ordinary skin cells can be transformed into cells that behave like eggs. This method has been tested in laboratories and shows that human life could potentially begin in ways previously thought impossible. The research not only demonstrates the power of science, but also challenges traditional ideas about reproduction. It has sparked global interest among scientists, doctors, and the public. Understanding how this process works and its potential impact is crucial. This discovery may change fertility treatments, reproductive rights, and even the way humans approach genetic health and family planning in the future. The science behind turning skin cells into eggs. The process of creating human eggs from skin cells relies on advanced cellular biology. Scientists start by taking ordinary skin cells and converting them into pluripotent stem cells, which can develop into any cell type in the body. These stem cells are then guided through a series of chemical and genetic signals that mimic natural egg development. Over time, they begin to form oocytes, the precursor to eggs. This process requires careful monitoring and precise laboratory conditions to ensure cells develop correctly. By replicating the natural stages of reproduction in a lab, researchers can study human development in a controlled way. The technique also allows scientists to explore the genetic and cellular factors that influence fertility. While still experimental, this method provides insight into human reproduction and could lead to revolutionary fertility solutions soon. How stem cells make this possible? Stem cells are the foundation of this breakthrough because of their unique ability to transform into specialized cells. By taking skin cells and reprogramming them into induced pluripotent stem cells, also known as iPSCs, scientists create a versatile cell type that can develop into eggs. These iPSCs are exposed to specific growth factors and signaling molecules that trigger their transformation into reproductive cells. The process closely mimics natural egg development in the human body, allowing the cells to grow, mature, and gain the potential to be fertilized. Stem cells also allow researchers to study the genetic stability and health of these eggs, which is critical for safe fertility applications. This approach is not only a breakthrough for reproductive science, but also highlights the potential of stem cells in regenerative medicine, genetic research, and personalized treatments. Potential impact on fertility treatments. This discovery could completely redefine fertility treatments around the world. For individuals who cannot produce eggs naturally due to age, medical conditions, or genetic factors, creating eggs from skin cells offers a new pathway to parenthood. It may also benefit same-sex couples, single parents, and people undergoing cancer treatments that affect reproductive cells. By generating eggs in the lab, doctors could reduce dependence on egg donors and improve success rates for in vitro fertilization. Furthermore, researchers can study egg quality, maturation, and genetic health in a controlled environment, which may help prevent certain genetic disorders. While clinical applications are still in the future, this technology has the potential to make fertility treatments more accessible, personalized, and effective, giving hope to millions of people who previously faced limited options. Ethical considerations and debates. The ability to create human eggs from skin cells raises significant ethical questions. Some concerns focus on the potential misuse of this technology, such as designer babies, genetic manipulation, or creating embryos for non-reproductive purposes. There are also questions about the rights of the future child and whether lab-created eggs may have unforeseen health risks. Religious and cultural groups may oppose altering natural reproduction, while scientists emphasize the potential benefits for fertility and genetic research. Another debate involves consent and ownership of cells used to create eggs, as well as long-term effects that are not yet fully understood. Balancing scientific progress with ethical responsibility is essential. Success stories in the lab. Recent experiments have shown promising results in creating human eggs from skin cells. In laboratory settings, scientists successfully transformed skin cells into pluripotent stem cells, which then developed into mature oocytes. Some of these lab-grown eggs were tested for quality and gene expression, showing remarkable similarity to natural eggs. researchers have documented successful maturation processes and preliminary fertilization experiments in animal models, demonstrating the potential of this technique. These achievements validate the concept that human eggs can be generated in controlled conditions, offering proof of concept for future clinical applications. Lab success also provides insight into reproductive biology, allowing scientists to study egg development, maturation, and fertility problems in detail. Possible implications for same-sex couples. Creating eggs from skin cells could open new opportunities for same-sex couples to have genetically related children. For female couples, lab-generated eggs could reduce reliance on donors, allowing both partners to contribute genetically. Male couples could also benefit if research progresses to creating functional eggs from male skin cells, though this is still experimental. This technology offers more reproductive freedom, enabling individuals and couples to have children without traditional biological limitations. It could also change adoption and fertility discussions, giving same-sex couples access to genetic parenthood. Additionally, studying lab-generated eggs can help scientists understand fertility barriers and improve reproductive options for everyone. Risks and scientific challenges ahead. Although promising, creating eggs from skin cells comes with significant risks and challenges. Lab-grown eggs may carry genetic or chromosomal abnormalities, which could affect embryo development. The process requires precise cell reprogramming and environmental control, making it technically complex. Long-term health effects of children born from lab-created eggs are unknown, raising safety concerns. Scientists also face challenges in replicating natural egg maturation and ensuring functional fertility outcomes. Ethical issues add complexity, as improper use could lead to controversial applications like designer babies. Regulatory approval will require extensive clinical trials and safety verification, which could take years. Overcoming these hurdles is essential before human applications. Future of human reproduction and genetic engineering. This breakthrough could revolutionize human reproduction and genetic engineering. Creating eggs from skin cells may allow for precise genetic screening, reducing inherited disorders. It could make fertility treatments more accessible and personalized, enabling individuals to have children later in life or overcome reproductive challenges. Scientists might also explore combining this technology with gene editing, potentially addressing conditions caused by faulty genes. Beyond fertility, this research may lead to advances in regenerative medicine, organ development, and understanding early human development. Society may see shifts in family planning, parenting options, and reproductive ethics as these techniques become viable. What this means for the future of life. Creating human eggs from skin cells marks a turning point in science with far-reaching consequences for the future of life. It offers the potential to overcome infertility, expand reproductive freedom, and even allow for new forms of genetic inheritance. Researchers can study human development in detail, helping to prevent genetic disorders and improve health outcomes. This technology could influence societal norms, family structures, and ethical frameworks as humans gain more control over reproduction. It also sparks debates on the limits of scientific intervention and the responsibilities that come with altering life's natural processes. As science progresses, humanity faces a future where reproduction is more flexible, informed, and technologically supported, raising profound questions about what it means to create and sustain life. This breakthrough could change the way we think about life and fertility forever. If you found this mind-blowing, hit like, subscribe, and turn on notifications to stay updated on the latest in science and innovation.